Hello, everyone. Thank you so much for joining us and welcome to iThemba Laboratory for Accelerator-Based Sciences as we showcase the accelerator infrastructure and expertise and a few of the nanoscience research projects conducted at our facility. All of this will be done for National Science Week 2021. My name is Hilton Tobias and I'm the Education Officer here at iThemba Labs. Now, let me tell you about iThemba Labs. iThemba Labs is a national research laboratory which hosts a number of machines called particle accelerators. Now, accelerators are big machines that make small things go really fast. I will not go into much detail now, but let's think about accelerators as the probes that allow us to look or peek inside atoms and the nucleus as we try to uncover the many mysteries of our universe. One of those mysteries is nanoscience and nanotechnology. Now, nanotechnology or nanoscience is defined by the Department of Science and Innovation as a process whereby one can manipulate the characteristics of materials at the atomic level, otherwise known as the nanoscale, which lies in the length scale under 100 nanometers. Now, nanotechnology has developed very rapidly since the dawn of the millennium and give rise to a new world of opportunities for developing countries such as South Africa. Let's look a bit deeper into the nanoscience in a different way. To do so, let's think about one single hair on the top of your head, just like this. And say we take that one piece of hair and we cut it 1 million times. Each small piece that we cut of the hair represents 1 nanometer, or in this case, 1 nanometer sized hair. So then to conceptualize it a bit more, it seems that if you can take 20 of these nano-sized hairs and you clump them together, the size of that will be about 20 nanometers. And 20 nanometers is about the size of the parvovirus. And if you clump more than 100, you get the size of the influenza virus. If you manage to put two of those tiny little nano hairs together, you get to the size of something called carbon nanowires, which displays a number of unique properties. So by extension, doing nanoscience research, where we are able to understand the different environments in which these novel materials can be developed and understand their characteristics, but also figure out how nanomaterials can assist in different areas. Today, we'll cover different applications of nanotechnology, like nanofluids, power generation units using solar receivers coated with some kind of nano coating, as well as the production of smart materials. So to start this journey into nanoscience or nanotechnology at iThemba Labs, we'll hear from Professor Malik Maza. Now, Professor Malik Maza is one of our research fellows here at iThemba Labs, and he currently holds the UNESCO African Chair for Nanoscience and Nanotechnology. He's also the principal investigator for the Nanoscience Research Group based at iThemba Labs. After the short talk of Professor Malik Maza, we'll be taking a guided virtual tour through the various experimental vaults here at iThemba Labs. And some of the research scientists will be talking to us about some of the amazing science that couples with nanoscience at iThemba Labs. We hope that this exhibition piques your interest in the science and makes you more curious about what's happening at the nanoscale. So one day you are able to make a difference in the world and not only the nano world. So I hope you enjoy it. Let's hear from Prof. Malik.
If I try to highlight some examples that you are working on, I would like to more or less to highlight this typical case of coatings that we are synthesizing. These coatings are smart materials. If we coat a window with such a material, when it's hot outside, the heat is reflected from the sun. And when it is cold, the heat is transmitted. So these coatings, once coated on a window of a car or a building, it acts like a smart air conditioning. Other materials on which we are working are materials for solar power technology, such as these ones, for example. As you can see, these coatings are black because they have a tendency and the property to absorb light from the sun and to transfer it in a form of heat. And these coatings are mainly used for the what we call CSP technology, this concentrated solar power, which is the other form of solar conversion parallel to the photovoltaics. Likewise, in these activities photonics, we synthesize and design and engineer materials which can be used to, such as this one, to purify water, contaminated wastewater. And these nanomaterials, once you disperse them in contaminated wastewater, you expose them to the sun, the water is purified and it's drinkable in effect. An additional activity that we are conducting, which is related also to energy sustainability, is what we call nanofluids. A nanofluid is a fluid which contains nanoparticles, metallic nanoparticles in general. And because a fluid such as this one, an oil, an engine oil, it has a low thermal conductivity. There's a huge loss of energy via heating, and therefore high consumption of fuel. The idea is to ensure that the thermal conductivity of the oil in the engine is higher, is enhanced, and so therefore to incorporate in it nanoparticles of metals, and generally speaking copper. By doing as such, we combine the viscosity of oils, which have a low thermal conductivity, as you can see here, to nanoparticles which have a high thermal conductivity. And therefore, by doing as such, we obtain a nanofluid, and doing as such, we reach high thermal conductivities, and therefore high efficiency, less loss in terms of heat in an engine, and therefore less consumption of fuel. This will be able to be translated to a marketable product and contributing therefore to an energy sustainability worldwide.
Hi guys, my name is Dr. Christopher Mjali from Ion Beam Analysis Division at the Tandertron section of iThemba Labs. I am going to take you through a tour within our section whereby we analyze materials using charged particles and also modify different materials using charged particles for different purposes.
Hi everyone, my name is Zakele Kumalo, and welcome to Material Research Department of iThemba Labs.
Good day everyone, my name is Dr. Nanotechnology. I'm a research scientist in the Ion Beam Analysis Group at iThemba Labs. Today I will be taking you through the virtual tour of our lab. Okay, so this here is the control room of the Tandertron Accelerator Lab, which is for the 3MD Tandertron Accelerator, which was commissioned and installed in 2016-2017. So this accelerator was commissioned with 2 ion sources for protons and alpha beams, as well as a cesium source for heavy ions.
So in terms of the light elements, we produce alphas as well as protons, and then with the maximum intensity of the protons being around 250 electrical microamp, and then one for alphas, the maximum intensity is about 25 electrical microamp. That is at maximum terminal voltage that you are using, because obviously we're using a terminal voltage to extract those charged particles that are being produced, just to accelerate them to extremely high speed. Okay, we also have the source for the heavy elements, which is called sputter source, which is on this side of the picture that is shown over there. So, and then the terminal voltage that we are using is positive. So that means the charged particles that are produced from these sources, which is this section, are negatively charged particles. So and then we are actually using electric field concept to actually do the acceleration of the charged particles, okay? So basically what you are seeing in this slide or in these pictures or in these computers that you're seeing here, we have a different softwares, because this is a control room, because everything is remotely controlled. So we have different software that control the production of your charged particles, going to the acceleration, determining the energies that you need for your experiment, or selecting the type of the charged particles that you are looking for when you're working with protons, for example. We have the parameters for that. And then we have, when I work with alphas as well, there are parameters that you can set on that software to be able to produce the type of the particles that you want to work, that you want to use for the experiment, okay? So that's basically just in this control room, that's where we sit and set up all the parameters that are necessary for the experiment that you will be doing on the day.
Okay, this here now is the Tandertron accelerator. Here is the cesium source that I talked about. And behind there, we have the two multicast ion sources for proton and alpha beams. So this accelerator accelerates ions in two steps. The first step is the extraction of negative ions from the ion source. So these ions are then analyzed and injected into the accelerator tank. So the negative ions get accelerated due to the positive high voltage potential that is applied here. So when these negative ions reach the terminal of the accelerator, there's ion exchange processes that take place. So these electrons, the negative ions, they get stripped of one or several of the electrons due to charge exchange processes that take place. So after this process, the beam now is composed of positive ions, which are then accelerated into the beam line.
So this section is called the high energy beam lines. So a different section, obviously, you need to monitor your current. So we have what is called Faraday curves that keeps on monitoring the current of the beam that we are accelerating. And then another field that we are using in this accelerator, it's a magnetic field. For example, if you see this big magnet, so it is there to actually bend your charged particles. So this is called 90-degree bending magnet. So what is happening, in fact, is that from this side, you have a cocktail of charged particles coming from here. So with different charge to mass ratio, so that means from the control room that I've shown in the previous slide, you set the right parameters to only bend the charged particles that you are looking for your experiment using this bending magnet. So the other charged particles with different charge to mass ratio then will be scattered within the magnet. So the only one that will be allowed to this beam line down to your experimental area, it will be only the one that you are looking for your experiment. And then you also have another components of this, which is actually what is called as steerers, that steers the beam, keep the beam at the center of the beam lines. And then from there it goes straight to your experimental area where you'll be performing your experiment.
So this here is the switch magnet, which is there to deliver beam to different beam lines. So at the moment we have two active beam lines, which is the solid state beam line that is dedicated to Rutherford backscattering spectroscopy, elastic recoil detection analysis, channeling, as well as sample irradiation.
So the techniques that you can use in this beamline, one of them is called RBS, in short, which is actually Rutherford backscattering spectrometry. And that's a technique that is actually based on the discovery of the nucleus by Ernest Rutherford. That's why it's called the Rutherford backscattering spectrometry. So the techniques one can use, for example, if you want to see what is in your material in terms of the elements, so you can use that techniques. It can tell you that you have, for example, you have your platinum, you have your gold, you have your copper, and so on and so forth. And then the interesting part, it can also tell you how much, for example, if you have those 2 elements present or 3 elements present in your samples, you'll be able to tell how much of gold as compared to your platinum or as compared to your gold. That's more like a stoichiometry or the composition of the material. So you can get that from these techniques, which is called Rutherford backscattering spectrometry.
So the principle of Rutherford backscattering is that incident alpha beam is impinged on the sample, and when this ion enter into the sample, there are a number of processes that take place. One of those is that some ions will go through, others will backscatter. So for Rutherford, we measure the backscattered ions. So by measuring the energy of these backscattered ions, we are able to determine the composition of element as well as their depth in the sample. So for elastic recoil detection analysis technique, ERDA in short, the theory is similar to that of RBS, but the difference now is in the experimental setup. For the Rutherford backscattering, the detector is placed in the backward position, and then for the ERDA, it is in the forward direction. And this is mostly used now for hydrogen storage material investigation. For RBS, it's thin films, you get the composition of filament and their depth.
We use this chamber here to analyze those paintings or those coatings. It is used for different applications, especially for electronics applications, whereby we use techniques such as Rutherford backscattering techniques to study the thicknesses of those paintings or coatings, and also the composition, which means the elements that are present in those coatings. And at the same time, this chamber here, we use it to modify the surfaces, either of those coatings or for a normal substrate or just the material that you are interested in. So we can create the nanoparticles on the surface of that material, or in those paintings, we can create nanoparticles by implanting or irradiating with the charged particle. When I'm talking about charged particles, I'm talking about the particles that you can generate from your periodic table elements, and you can implant them in your material. And you change, by implanting them, you change the properties of your material, such as a typical example of your VO2, the vanadium dioxide coating, which is got a potential of being used as a smart window, whereby you can change the properties in such a way that at a particular temperature, you can be able to block the sunlight or reflect sunlight. But at a particular temperature, let's say below 30 degrees, you can be able to allow the sunlight to go inside your room, and therefore warming up your room. But above that, you can block it in such a way that you keep your room at a very favorable temperature. So there's another typical example that we use this chamber for.
Okay, now we move on to our another beamline, which is the one which we use to carry out particle-induced X-ray emission spectropixy. So the beam will travel through this beamline, and before the beam reaches now the target chamber where we place our sample. It passes through these coordinators, which define the beam and focuses it. And then this here we have the quadrupole lenses, which are there now to focus the beam. Once the proton beam interact with your sample, these ions, they have enough energy now to knock off the electrons in the sample atoms. So when the electrons get excited, they will be ejected from the inner energy shell. So another electron from a higher energy level, because now the atom is unstable, would want to now move to that vacancy. And when it does that, it loses energy in the form of X-rays. So these X-rays are the ones that are detected, and they carry information regarding the elements that are present in your material. So the detector that we use to detect these X-rays is this silicon lithium detector, which is this one here. So when the X-rays are detected, signal is sent from the preamplifier to the amplifier and then to the analog to digital converter to eventually produce the energy pixel spectrum. So now Pixie, the example of projects that we have conducted using this is to study biological samples. What we did is we investigated hyper-accumulating plants. So we were able to find out the amount of nickel in those plants as well as their distribution. Another project is where we studied the human hair, which is for forensic studies. So if a person is poisoned, you can just take the strand of the hair and then use Pixie to find out exactly if there are toxic materials in that sample. The other sample that we did are the fish scales. So we collected fish scales from contaminated ocean and then they analyzed the scale to find out which elements are present in that sample. So this is basically particle induced X-ray emission, elemental composition and distribution.
These techniques that I've just highlighted is a signal that is generated and then it needs to be processed and go to your computer for you to be able to sit down and interpret the spectrum and be able to tell all this information that I've just been highlighting, to tell what is in that sample and how much of it, and then actually in terms of the depth, where is it sitting in the samples that you have actually analyzed.
We don't analyze water. The only way to analyze samples such as water is whereby we use soil that is extracted from that water, and we'll be able to quantify and qualitatively analyze soil that is coming from that water, and we can relate to the elements that are present in your water samples. That's how we analyze water samples. Other than that, we analyze all types of samples, biological samples, your leaves, your roots, your stem of your your tree, in order to investigate the presence of either heavy elements in your tree or in your food, which are quite toxic to your body. And that is what we basically do in this. We run different projects, internally and external projects, which are based on what I've just described. Thank you very much. I hope you enjoy the talk and you will be able to engage us moving forward and keep on visiting our page. Thank you very much.
I hope that you enjoyed it. But in the near future, please feel free to come and visit us once all this restriction has been lifted. So once again, thank you for your attention.
I thank you so much for your attention, and hopefully you will be interested in joining our lab. Thank you so much.
So this brings us to the end of our virtual tour exhibition for National Science Week. If you're interested in Taking a look around the virtual platform, you can find it on our website at www.tlabs.ac.za. If you would like to get in contact with us, you can email us on director@tlabs.ac.za. Otherwise, follow us on our social media with the Facebook handles at iThemba SE and at iThemba Labs Cape. Thank you so much. We hope you enjoyed it.